Okay, anatomically, parts of the I will be quick. I don't know if I'm going to write it down or not. Parts of the nervous system, central, peripheral, right? Know the functions, generally functions. Be able to tell them apart, which function is for which one. Okay? Anatomical structures that comprise, so CNS, brain, brain stem, spinal cord, peripheral nerves and receptors. Anatomical part, I gave you the anatomical part, know the functions. Okay? We talked about them extensively. If I ask you which of the following parts is for thinking, you tell me it's the cortex or cerebrum. Okay? Are we clear? Cells, know the functions. Okay? This is what we call declarative memory. It's not procedural. Okay? Answer all those questions. 4 types of glial cells in the CNS. Know that Schwann cells and oligodendrocytes are functionally same, but Schwann cells are in the PNS. Okay? Clear? Function of myelin, electrical insulation, disease in which... malignancies, I mean cancers, it's in the table, like neuroma, okay, or microglioma, or astrocytoma. They kind of self-explanatory, okay? Disease in which immune system attacks myelin sheath is MS. Major parts of neuron. Look, we went over this a million times. So, functions of those parts, reception or transmission of the signal, sequence of parts through which signal goes through. Okay? 3 types of neurons. Multipolar, bipolar, unipolar. I give you description, you give me the answer. Are we clear? You've got the question about it. Synapse. Place where 2 cells transmit signal, one to another. Okay? Signal can be amplified, inhibited, unchanged, changed, blah, blah, blah. 2 types of the synapses, chemical and electrical. Know the difference. Okay? 1 direction, 2 directions. Neurotransmitter is released or ionic flow through the gap junctions. Where you can find them, chemical synapses, dominant in the CNS, electrical synapses between the smooth muscle cells and cardiac muscle cells. Steps of the synaptic transmission. I'm not going to rehearse it. We have 1 slide. It's 2. 1 slide with the synapse. And we went over this. Okay? Action potential, calcium influx, release of neurotransmitter. I'm skipping on some of them. Please know it in exact sequence. I will test you on that. Okay? What neuron is presynaptic? What is postsynaptic? What's neurotransmitter? What is the synaptic cleft, the space between those neurons? Role of calcium. Neurotransmitter is released in response to calcium influx. How it happens? Exocytosis, fusion of vesicles to the membrane. What happens to the neurotransmitter after interaction with the receptors? It's getting degraded. Know how it is getting degraded. Does the transmission require energy? Yes, it does. Types of receptors. Ionotropic and metabotropic. Okay? Differences between them. Ionotropic opens ion channels. Metabotropic, second messengers. Know that, please. We clear? We good? I'm going fast. It's all getting recorded. It's getting recorded good. It's all going to be on YouTube. Okay? what is excitation, what's inhibition? Like excitatory neurotransmitter, sodium in. Inhibitory neurotransmitter, chloride in or potassium out. Does that make sense? Second messenger, G protein activates intracellular signaling pathways. G protein that's inside of the cell. Okay? Two types of neurotransmitters, neuropeptides and small molecules. Okay? Small molecules fast, short-lived, neuropeptides slow, long-lasting effects. Okay, this. You have a table. Know it. There are going to be questions about them. When I say what is agonist and antagonist, when we say something is agonist for a receptor, what does that mean? It stimulates. Antagonist inhibits the signaling. Does it make sense? So serotonin receptor agonist will stimulate serotonin signaling. Serotonin receptor antagonist will inhibit serotonin signaling. Does that make sense? Please understand what happens if you inhibit receptor. When you inhibit the reuptake, when you inhibit the degradation, when you stimulate the receptors, essentially these three will increase the signal. Okay? This one will decrease the signal. How effects can be used. Remember the question about cocaine? Inhibition of reuptake, how can you counteract this? You're going to get a question like that. I promise. Okay? Essentially, it gives you a space to go over each neurotransmitter. ACH, I'm going to give you an example on, say, acetylcholine, okay? ACH, it's either excitatory or inhibitory. Organophosphates inhibit degradation. Botulism inhibits release. Atropine inhibits receptors, okay? What happens in myasthenia gravis? Receptors are destroyed. Now, I want to be very clear. Please don't waste time and memorize the effects of, say, botulism toxin. But if I tell you in myasthenia gravis, receptors for ACH are destroyed, you have to understand what are the consequences of it. Does that make sense? I tell you, organophosphates inhibit ACH degradation. What's going to happen to the signaling? It will increase because higher level of ACH, right? So don't waste time memorizing the stuff like what does Prozac inhibit. Okay? But think, okay, he says that Prozac inhibits serotonin uptake. How does it change serotonin signal? Am I clear? Focus on this rather than on direct effect. So I'm going to skip the list of these guys. Okay? I'm going to have glycine twice. Oh, it's nitric oxide here. Yeah, it should be nitric oxide. So it pretty much same goes for neuropeptides. Don't focus that much on like specific. So if I tell you opiates interact with the same receptors as endocannabinoids, okay, and stimulate the same stuff. Are they antagonists or agonists? They're agonists. This table, I hope, self-explanatory. If I tell you, you open such and such channels, what's going to happen to the membrane potential? I presume that you know where the ion goes into or out. Does that make sense? So if I tell you I open potassium channels, you remember potassium is higher inside, it's going to flow out. Okay? Types of ion channels: leaky, mechanically gated, ligand gated, voltage gated. No ATP required. Know what controls each channel. Nernst potential. How it is formed, what it is. Net flow is 0, right? What's going to happen to Nernst potential if you say increase or decrease the number of channels? So I can tell you, if for potassium I will increase the number of potassium channels in the membrane, what's going to happen to Nernst potential? It's going to become even more negative because there's going to be more potassium flowing out. If I will increase the number of channels for sodium, it's going to become even more positive because more sodium will flow in. Does that make sense? Have an idea about that? RMP, understand how it is formed. Potassium out, sodium in, sodium-potassium pump restores the membrane. Depolarization, hyperpolarization, which ions can do. Okay? Know which way the ions go. Graded potential, know the difference between graded and action potential. The graded potential, if it doesn't cross the threshold, it will dissipate, it will go away. When it crosses the threshold, it becomes action potential. Okay? Ionic flow. That graph. Depolarization, repolarization, hyperpolarization. Which ions are responsible for each step? What is absolute refractory period and relative refractory period? Okay? Understand the idea of all or none. There's no difference in value, only frequency. Here, potassium channels are slow, that's why hyperpolarization. Absolute refractory period, well, covered it. Electrical impulse travel through non-myelinated axon. Remember, opening the channels. Myelinated, same, but between nodes of Ranvier. Saltatory conduction. Okay, ion channels open in the nodes of Ranvier. Again, excitatory, inhibitory, postsynaptic potential. Excitatory opening of sodium channels, inhibitory opening of chloride or potassium channels. What happens to the membrane potential in each case? Summation. Understand the difference between spatial and temporal. There will be a question about that. Okay? Sorry. Discharge versus facilitated zone for the neuronal pool. We had this question in the quiz, something that sort is going to be. Types of neuronal circuits. I give you the function, you tell me the circuit. Okay? Sensory information, convergent. Okay? Motor output, divergent. Does that make sense? That's going to be pretty straightforward. This is brutal memorization. First, primary embryonic vesicles, secondary embryonic vesicles. What do they develop into? I'm not going to go over it. It's all in the first slide of the brain stuff. Okay? Just like telencephalon becomes cerebrum. Okay? Mesencephalon becomes midbrain. Same goes for the ventricles. Anatomy of ventricles. Know it. Horns of the lateral, where is the third, where is cerebral aqueduct, where is the foramen, where is the fourth ventricle, where is central canal, and stuff like that. Function of CSF, produced by choroid plexus, drained through the arachnoid villi into the blood. What's hydrocephalus? We drain the excessive fluid into the abdomen. Three meninges. Order, anatomically, recognize them. Spaces, subdural, subarachnoid. What's in those spaces? Subarachnoid, CSF, and large blood vessels. Okay? So this subarachnoid space, arachnoid villi. Arachnoid villi are formed from arachnoid matter. Separation of periosteal and meningeal layers of dura mater gives you venous sinuses. There was a question on the quiz. Anatomy of the brain, hemispheres, left and right. If you accidentally say that this is not the left hemisphere but right hemisphere, it's a wrong answer. Gyri, sulci, median, median sagittal fissure, transverse cerebral fissure, lobes. Okay? Clear? Parts of the cerebral. Major parts of the cerebrum. Cortex, basal nuclei, white matter. General functions. Understand what is functional MRI. What does it do? Regions of the motor cortex. Now, since we limited in time, for motor cortex, know the functions of things like premotor versus motor cortex. Somatosensory, function, for all of them, no location. Again, what do I mean by that? I point here and say, which lobe is that and what type of cortex you can find there. Okay? So which lobe is that and what type of cortex? Occipital visual cortex. This type of question you can encounter on both 50 questioning, lecture exam. On anatomy exam, you're going to have a picture. I show you this, and you tell me lobe, and you pick proper combination of the lobe and the cortex. Okay? Okay, I'm skipping. Skipping. Somatotopy. Understand both motor and sensory, size of the body to the size of, ability to do fine motor activities corresponds to the size of the cortex, and the number of sensory receptors corresponds to the size of the cortex. Lateralization of the functions, decussation. White matter, three types of fibers, self-explanatory. Basal nuclei, function, mostly inhibition of motor activities. Pathologies, Parkinson's and Huntington's. Thalamus, relay. Hypothalamus, visceral controls, visceral response to emotions. Three parts of brainstem with the functions that we discussed. It kind of tells you what to know. Corpora quadrigemina, visual startle reflexes, Substantia, what? Wait. Oh, I screwed up. Oh, it should have been Pons. I have no idea how it, oh, I know. I was trying to do something like, copy paste, and it turned into C. For Pons. Respiratory center and the bridge between the cerebellum and the cortices. Okay? Medulla functions identified on the pictures. Cerebellum functions also know the flow of information. Does that make sense? Remember today there was a question, what type of sensory inputs does it receive? Cerebellum? Have an idea about that? Visual, vestibular, proprioception, and planned movements. Okay? There you got it. It kind of linked. Okay? Cerebellum communicates with the cortex, with the peripheral receptors, but does not control muscle activity directly. Limbic system, emotional responses, including visceral responses to emotions. Reticular formation, alertness. Okay? Body functions. Consciousness, we talked about three criteria of consciousness, 4 levels. Fainting, coma. Know the difference between fainting, coma, and sleep. 4 brain waves, what they correspond to. 5 stages of sleep. Muscle inhibition during REM sleep. Sleep disorders. Memory, long-term versus short-term, procedural versus declarative. The pathologist, traumatic brain injury, ischemic stroke, hemorrhagic stroke. Know the difference. Alzheimer's, Parkinson's, Huntington's, prions. Be able to recognize the disease by the symptoms, that's number 1. Second, in Parkinson's, remember there's a dopamine stuff. I will give you an introduction, okay? It's like there are problems with dopamine production. What would you use to treat it, okay? Deterioration of this too. Prions, spinal cord, what we covered today. Please focus on the pathways. That's the hardest part, really. Okay? Focus on the pathways. Since we are out of time, okay? Anatomically, what we discussed today, focus on the pathways. Anatomy and functions. Okay, in the meantime, email me.